Hello students, welcome to e-learning classes. I am Piali Nag, your biology teacher, and I will be taking your next chapters of the biology. Okay, so let's get started. We will start with the Human Genome Project. Okay, now as the name suggests, the Human Genome Project deals with the whole study of whole genome content inside the human beings. This includes the sequencing or knowing the sequence of the gene inside the DNA of every gene, and this was a huge project which was carried out by the scientists, and the total cost of the project was around 9 billion US dollars, and an enormous amount of data was generated from this project, so it is also called a mega project. Okay, now the Human Genome Project was a 13-year project by the US Department of Energy and the National Institute of Health. It was launched in 1990 and was completed in 2003. But during the early years of the Human Genome Project, the Wellcome Trust from the UK became a major partner, and the additional contributions came from Japan, France, Germany and China. The Human Genome Project was actually closely related with the development of a new area in biology, which was called the bioinformatics. Now, what are the basic goals of the Human Genome Project? The basic goals included to identify 20,000 to 25,000 genes in the human DNA, to determine all the 3 billion chemical base pair sequences that make up human DNA, to store the above information in the databases, to improve the tools for the data analysis, to transfer the technologies to other sectors such as industries, etc., and to address the LZ, that is the ethical, legal and the social issues which may arise from this mega project. Okay, now what are the advantages of the Human Genome Project? The effect of DNA variation which can be studied among the individuals and which can lead to the revolutionary new ways to diagnose and treat many disorders or diseases. This was the first and foremost advantage of HGP, and this also provided clues to understand the human biology. Moreover, more information can also be obtained about the non-human organisms like bacteria, yeast, nematode, fruit fly, plant, rice, etc., which were very much similar, which contain very much similar genes from similar genes like the humans. Okay, now what were the methodologies of the Human Genome Project? There were two major approaches of the Human Genome Project. The first one is the Express Sequence Tags, that is the ESTs. This method focuses on identifying all the genes that are expressed in the RNA. That means from the translation process, the genes which were translated to the proteins, that to identify all these sequences. Alright, and the second methodology was the sequence annotation, which was an important approach of simply sequencing the whole, means rearranging the whole genes according to the order it is present. Okay, that means which contained all the coding and non-coding sequences, and later assigning different regions in the sequences with their particular function. Now, for sequencing, the first total DNA from the cell was isolated and broken down relatively into smaller fragments. Now, these DNA fragments were then cloned in suitable hosts using suitable Vectors. You will come to know what are vectors in the biotechnology chapters, right? Next is when the bacteria was used as a vector, they were called the bacterial artificial chromosomes. And when yeast is used as a vector, they were called the yeast artificial chromosomes. Basically, Frederick Sanger developed a principle according to which the fragments of the DNA were sequenced by automated DNA sequences, and on the basis of overlapping regions which are in the DNA fragments, these sequences were arranged accordingly. Now, for the alignment of these sequences, specialized computer-based programs were also developed, and finally the genetic and the physical maps of the genome were constructed by collecting information about certain repetitive DNA sequences. The repetitive DNA sequence, actually the whole DNA consists of few repetitive sequences. These are actually non-genetic part of the DNA, and this repetitive sequences makes a person unique, that means different from each other. These are unique in every organism except the clones, okay? And DNA polymerization, which was based on the endonuclease recognition sites. Now, coming to the salient features of the Human Genome Project, actually the human genome consists of 3164.7 million nucleotide bases, and the average gene was 3000 bases. So the largest known human gene being dystrophin at 2.4 million bases. The total number of genes is estimated to be 30,000 and 99.9% nucleotide bases, which were exactly the same in all people. Okay, now the functions are unknown for over 50% of the discovered genes, and less than 2% of the genome coded for proteins. The rest were non-coding genes. And the human genes also contain large repetitive sequence, which I have already told you. These repetitive sequence are also called as satellite, that is the microsatellite, mini satellite, then there are VNTRs. These are all repetitive sequences, and these are termed as mini and micro according to their length. Okay, now the repetitive sequence is thought to have no direct coding function, but they throw light on the chromosome structures, dynamics and evolution. Like chromosome 1 has most of the genes, the 2968 genes are present in chromosome 1, and this was discovered by the while ongoing the Human Genome Project. And the Y chromosome, which actually determine the sex of the males, this contains the most less number of genes. Okay. And the scientists also had 1.4 million locations where single base DNA sequences like the single nucleotide polymorphism occurred in humans. What were the applications and future challenges for the HGP? The complete sequence of human genome, when properly studied, was enabled to develop radically new approach to the biological research. From here only we can think of sequence changing, that is the artificial mutation which were carried out by the scientists or the human beings. Okay, we can think of what were how these mutation can be carried out. Carrying out the mutation reactions was much more easier, since the sequence of the humans were already discovered by this project. Okay, and then
All the genes in a genome and transcripts in a particular tissue, organ or tumor were studied. And it also enabled us to understand how enormous number of genes and proteins work together. So today up to this, we will continue with the rest part of the chapter in my next videos. Thank you.